Welcome to the problem-solving set on DNA replication. Question 1 is asking us to compare DNA replication in prokaryotes versus eukaryotes. Now I know that Professor Schmidt went through this in detail in the theory video, so let's just do an overview of the basic differences. So in prokaryotes, or bacteria, their DNA is circular. So they have circular DNA. And this means that they only need one origin of replication. So they have one. In terms of DNA polymerase, they have 5 different DNA polymerases. And they also have another enzyme called topoisomerases. And these relieve the pressure of supercoiling, which happens when the replication of a circular piece of DNA happens. So relieves pressure of supercoiling. Okay. In contrast, eukaryotes, which obviously humans are an example of, we have linear DNA that's very long, which means that we must have multiple origin of replications. We have at least 7 different DNA polymerases. And we also have an enzyme called DNA telomerase, and that replenishes the telomeres that are on the end of our DNA. So replenishes Okay, so if you want more detail comparing the DNA between prokaryotes and eukaryotes, feel free to rewatch the theory video, or at least just look through the notes. But I think these are the most obvious and simple differences between prokaryotes and eukaryotes. Retroviruses carry their genetic material as RNA that is used as a template for DNA that is then incorporated into the infected cell's genome. For the section of viral RNA shown as a template, what would the sequence of the first strand of DNA made? Okay, so viruses that use RNA as their genetic material also carry an enzyme known as reverse transcriptase. And this enzyme uses an RNA template and makes a DNA that goes into the nucleus of the cell, and then actually incorporates into the infected cell's genome. So what should we remember about RNA? They use all the same bases except that thymine is replaced with uracil, so all these U's in DNA would be T's. However, because this is a template, we're going to use the same anti-parallel rules and the same rules for base pairing. The only thing is that here the U's are going to be base pairing with A's. So if we're starting with 3' on the template strand, the new strand is going to be 3', then G, G. Remember that U's replace thymines in RNA, so they will base pair with A's. Okay, then C, T, T. Remember we're using T because now we're making a DNA strand. C, A, G, C, G, A, T, T, G, C, A, A. And that's 5'. So this is the strand of DNA that's made from using this RNA strand as a template. Question 3. An origin of replication is located within the region of DNA shown below. Determine the sequence of the primer that would be synthesized complementary to the bases in blue, indicating the polarity of the sequence. What enzyme is responsible for the synthesis? Now remember, for DNA replication, which is done through the DNA polymerase, that DNA polymerase can't initiate the synthesis of a strand by itself. It can only add to a free 3' end, which means that it needs a primer. Now primer is actually a short sequence of RNA, and this is done by the enzyme called DNA primase. Okay, now because DNA polymerase can only make new strands in the 5' to 3' direction, that means that it has to read the template strand in the 3' to 5' direction. So let's do the complementary primer to these bases in blue. So this is the 3' end on the right, so we're going to start with the 5' end. And remember also that this is an RNA primer. So C and G bind together still. However, an A here doesn't bind with a T because the primer is made up of RNA. So the A would bind with a U instead. T still binds with A, U, U, and then 3'. So DNA polymerase would, let's draw the enzyme like this. It would recognize this primer and then keep making a new strand in the 5' to 3' direction. Okay, so and just remember too that this is a little primer of RNA. And that's how you do the primer. Question 4. Regions of eukaryotic DNA replication appear as bubbles under the electron microscope. Use the diagram below to answer the following. How many origins of replication are active in this molecule? Assume bidirectional replication. So remember that origins of replication in eukaryotic DNA make the appearance of a replication bubble. So we can see 3 bubbles here. And the origins of replication, I'm going to draw as a line in blue, would be in the middle of all 3 of these replication bubbles. So there are 3 origin of replications active in this molecule. Now it's asking how many replication forks are present. Now remember, from each origin of replication, if it's somewhere in the middle of a DNA helix, that there is a fork of replication on either side. So there's a fork here and here, so that's 2, then 3 and 4, 5 and 6. So these are the forks from the replication bubble. So there are 6 forks present. Now it's saying which origin of replication was activated first and last. Assume all replication forks are synthesizing DNA at the same speed. So if DNA polymerase is, and then remember that they can have multiple copies of this enzyme. So if 3 enzyme DNA polymerases are going at the same speed in these replication bubbles, how can you tell which one was activated first? Well, the one that was activated first has been going the longest, which means that it has unwound the DNA double helix for a greater portion of the DNA. So this middle one was activated first, because you can see the replication bubble is longer. Now by that same logic, the one that was activated last was the most recent one, which means it hasn't unwound the DNA very far at all, which means this one on the left was activated last. And therefore the one on the right was activated sometime in the middle, so activated second. Problem 5. For the following statements about DNA synthesis in eukaryotes, determine if they hold true for the lagging strand, leading strand, both strands, or neither strand. For A, DNA synthesis is discontinuous. Well, that is actually a property of the lagging strand. And remember, it's discontinuous and it makes Okazaki fragments because it's going in the wrong polarity. For B, synthesis occurs in the 3' to 5' direction. Remember that DNA polymerase, which is what is responsible for DNA replication, reads the template strand in the 3' to 5' direction, but synthesis of new strands always has to occur in the 5' to 3' direction. So B is false for either strand, so let's say neither strand. C. DNA polymerase uses the rules of complementary base pairing when synthesizing the new strand. Well, that is true for both. And these complementary base pairing rules are that A base pairs with T and G base pairs with C. D. Synthesis produces Okazaki fragments that are eventually joined together by DNA ligase. Again, as we said in A, this is a property of the lagging strand. For E and F, it's asking which of the two where elongation progresses away from the replication fork or toward the replication fork. So what I want to do is draw a replication fork. We'll do just single directional replication, so let's do one fork. Okay, so this would be the origin here and a line down. And we're going to say that replication is happening in this direction to the right. Okay, so let's say that this top strand is 5' to 3', which means that the bottom strand is 3' to 5'. Now again, DNA polymerase reads the template strand from 3' to 5' and creates a new strand in the 5' to 3' direction. So the leading strand is going to be from the template on the bottom, and it'll be 5' to 3'. And for that strand, the elongation is to the right. The lagging strand, again, the new strand has to be made 5' to 3', so it's going to make small Okazaki fragments in the 5' to 3' direction. And as you can see, because elongation happens from 5' to 3' on the new strand, for this lagging strand, elongation is happening to the left. So which one is progressing away from the replication fork? If replication is happening toward the right and the replication fork is on the right, the one that's having elongation to the left is the lagging strand, so that is progressing away from the replication fork. Now it's asking which one has the elongation progressing toward the replication fork, and that is the leading strand. Finally, the last statement says, synthesis requires a template strand and a single-stranded RNA primer. Now again, this is true for both strands. So if we're going to draw the primer in, let's use green. For the leading strand, it's just going to need one RNA primer. It's single-stranded, and remember that that primer is made from DNA primase. However, for the Okazaki fragment, they need a primer on all of these fragments, because DNA polymerase cannot initiate the synthesis of a strand. It can only add bases to the 3' end. So after all the Okazaki fragments are synthesized, the primers are removed and DNA ligase attaches them together. So these are just some simple properties of both strands, and it's important that you get these down. Problem 6. If the replication fork starts at the left of the DNA sample shown below and moves toward the right, what is the sequence of DNA synthesized as the lagging strand? So if the replication fork is starting to the left and moves toward the right, that means that DNA replication is happening from left to right. Okay, now both leading and lagging strands are going to be synthesized in the 5' to 3' direction, because remember, DNA polymerase can only create new strands in this direction. Now the leading strand is going to have the template strand that is in the 3' to 5' direction, which is the direction that DNA polymerase reads, but the lagging strand is going to have the template that's in the 5' to 3' direction. So this top strand, that is going to be the template for the lagging strand. And that's because, again, it is 5' to 3'. Okay? So in order to get the sequence of DNA synthesized as the lagging strand, we're going to have to find the complementary of that. So as you can see, the complementary strand of that strand is actually already written for us, because DNA replication is just making another strand of complementary DNA. So we can copy it like that, so it's just copying the complementary strand. Okay, and that's in the 3' to 5' direction. But sometimes if you're on a test and there's only multiple choice, they might ask for it in the 5' to 3' direction. So all you're going to do is copy it backwards. So it's C, G, C, G, T, A, G, G, C, A, T. Okay, so either way of writing it is the same DNA strand. Just make sure if you're going to switch the polarity that you do keep track of the polarities. Question 7. The melting temperature of DNA is the temperature at which the double helix dissociates into single strands. Which of the following sequences would have the highest melting temperature, or in other words, would need the most energy to unwind? Explain. So when we look at these 3 sequences, now remember they're only showing one strand out of a double-stranded molecule. We're looking at them, you see that they all have the same amount of nucleotides. And what's really different about nucleotides? The only thing that varies between each nucleotide is its nitrogenous base. So when you're looking at these, the one thing that you're going to keep in mind that can change the amount of energy the temperature would need is that the A and T pair has only 2 hydrogen bonds, right? So A and T have 2 hydrogen bonds between them when they base pair, and G and C have 3 hydrogen bonds between them when they base pair. So what we're going to do is look at each and see which has the highest GC content. So let's count the number of G's and C's. In A, in one strand, they have GG 2, CCC 3, 4, 5. There's 5 G's and C's. In B, they only have 2 C's. And in C, they only have 1 G. So right now we're saying that A has the highest GC content. And this means that it's going to have the highest melting temperature. And the reason for that is because it has the highest GC content out of these 3 DNA strands, it's going to have the most amount of bonds because there's one more bond per the GC pairing than there is for the AT pairing, and therefore would need more energy to break them, so it has the higher melting temperature. Now, if you want to rank them, even though they don't ask us, the highest to lowest temperatures, A again is going to have the highest, which would be greater than B, which would be greater than C. And remember, that's because B has 2 C's, whereas C only has 1. So again, the higher the GC content, the higher the melting temperature is going to have to be because of that extra hydrogen bond.